Hey guys, welcome to SpecTransfer and to topic 3.2.1.3, methods of studying cells from the AQA A-level Biology specification. As always, let's start with a look at our specification. We should know about the principles and limitations of optical microscopes, as well as transmission and scanning electron microscopes. We should know how to measure the size of an object viewed with an optical microscope, as well as the difference between magnification and resolution. We should know that magnification equals the size of an image divided by the size of the real object. And finally, we should know about the principles of cell fractionation and ultracentrifugation and how they are used to separate cell components. We should also be able to appreciate that there was a considerable period of time during which the scientific community distinguished between artifacts and cell organelles. So let's make a start. First of all, we will compare light microscopes with electron microscopes. Light microscopes use a beam of light, whilst electron microscopes use a beam of electrons. Light microscopes have a lower magnification and resolving power, with a maximum magnification of times 1500 and a maximum resolution of 0.2 micrometers. Therefore, we cannot see ribosomes, endoplasmic reticula, and lysosomes using a light microscope. However, a benefit of light microscopes is that they can be used for live specimens. Electron microscopes, on the other hand, have a higher magnification and resolving power, with a maximum magnification of 1,500,000 and a maximum resolution of 0.002 micrometers. If asked in an exam why electron microscopes have a better resolution than light microscopes, you should state that electrons have a shorter wavelength than light. A disadvantage of electron microscopes, however, is that they cannot be used for live specimens, as the specimen has to be contained in a vacuum, as otherwise air particles would absorb electrons and interfere with the image generated. Note that there are two types of electron microscopes, transmission and scanning electron microscopes. In transmission electron microscopes, electrons are transmitted through the specimen. Denser parts of the specimen absorb more electrons and thus appear darker on the image. They can be used to visualize the internal structure of organelles. They have a higher resolution than scanning electron microscopes, but can only be used for thin specimens. Transmission electron microscopes generate a 2D image. Scanning electron microscopes, on the other hand, work by scanning a beam of electrons across a specimen. Electrons are knocked off the specimen and are collected in a cathode ray tube. Scanning electron microscopes only show a specimen's surface. Scanning electron microscopes have a lower resolution than transmission electron microscopes, and they can be used for thick specimens and generate a 3D image. Next, we need to learn a formula and two definitions. Magnification equals the image size over the actual size. I always found formula triangles useful when revising for exams, so I've included one here. The formula is easy to remember as it spells I am. We also need to know the definitions for resolution and magnification. Resolution is the minimum distance that two objects can be apart for them to appear as separate objects. Magnification is how many times the image is larger than the specimen. It's important not to get confused between resolution and magnification. Finally, we need to know about how cell components can be separated using cell fractionation and ultracentrifugation. Cell fractionation is the process by which cells are broken up and their organelles are separated out. The first step in cell fractionation is homogenization, which involves breaking up cells using a homogenizer, which is a blender, or by vibrating them. This breaks the plasma membrane of cells, and the organelles are released into the solution the cells were kept in. The solution cells are kept in must be cold to reduce enzyme activity, which may hydrolyze organelles. The solution must also be isotonic, which means that it has the same water potential as the cells. This prevents organelles from bursting or shrinking due to osmosis. And finally, the solution must contain a buffer to maintain a constant pH. A fluctuating pH could damage organelles or affect the functioning of enzymes. The second step in cell fractionation is filtration. In this step, the resultant fluid from the homogenization process, which is called the homogenate, is filtered to remove any unbroken cells or cell debris. Finally, we have ultracentrifugation. This is the process by which the different organelles in the filtered solution are separated out in a centrifuge. The mixture is first poured into a test tube. The tube is put into a centrifuge. This is then spun at a low speed. The heaviest organelles, such as the nuclei, are forced to the bottom and collect to form something known as the pellet. The suspension above the pellet is known as the supernatant, which is drained off, poured into another test tube, and spun at a higher speed. This time, lighter organelles, such as mitochondria, are found in the pellet. The supernatant is then drained off again, and the process is repeated. Each time the tube is spun at a higher speed, lighter and less dense organelles are forced to the bottom and are found in the pellet. The order in which organelles are found in the pellet is shown here, i.e. when we spin at low speeds, nuclei are found in the pellet. Then, in the next round of centrifugation, when we spin at a higher speed, mitochondria are found in the pellet, and so on. As you spin at a higher speed, lighter and less dense organelles are found in the pellet. Great, that would be methods of studying cells covered. We have covered the principles and limitations of optical microscopes, as well as transmission and scanning electron microscopes. We know how to measure the size of an object viewed with an optical microscope, as well as the difference between magnification and resolution. We have also covered that magnification equals the size of an image divided by the size of the real object. And finally, we covered the principles of cell fractionation and ultracentrifugation and how they are used to separate cell components. That would be it for now, guys. Thanks for watching. Please subscribe, comment. Next time, we will be covering topic 3.2.2, all cells arise from other cells.